start with the new topic, that is eukaryotes. So we know that there are two types of cells. First, eukaryotic cells, and second, prokaryotic cells. So let us begin with eukaryotic cells. So eukaryotic cells are the types of cells which are having so many multiple organelles inside them. Okay, they are the advanced cell. They are having advanced features than prokaryotic cell. So eukaryotic includes animal cell and plant cell. Now let us understand some of the basic things that are needed, which are always present in these two types of cells. There is a huge difference between animal cell and plant cell, but here are some basic information about it. First, cytoplasm. What is cytoplasm basically? So cytoplasm is nothing but it is a fluid content enclosed by the cell membrane. Fluid content, here you can see it is blue in color and here it is green in color. So this is known as what? This is known as cytoplasm. It contains specialized organelles. Organelles means organs, like here we are having human organs inside our whole body, like we are having organs, liver, stomach, understand, and all. So here there are some organelles inside these cells, and they are like this: mitochondria, endoplasmic reticulum, Golgi bodies, then plastids, ribosomes, here endoplasmic reticulum, mitochondria, Golgi bodies, plastids. No, plastids are not present here, but these all are organelles, the small specialized part which are present inside the cytoplasm. Organelles perform a specific function and are enclosed by the membrane. They are having special functions inside them. They perform a specific activity and they are enclosed by a membrane. Okay. Cell organelles. So cell organelles, eukaryotic cell contains various membrane bound organelles that carry out various functions. So eukaryotic cell only contains some membrane bound organelles. Just now we see, like what are the examples, like nucleus, endoplasmic reticulum, Golgi apparatus, and lysosomes. But there are several other things also. Okay, there are several other things as well. Next, here's one question. The only cell organelle that is found in prokaryotic cell is. So till now I haven't explained you about the organelles and all. So the only organelle which is found in prokaryotic cell is ribosomes, because prokaryotic cell is not having much specialized things, so it is only having ribosomes inside it. Okay. Next, endoplasmic reticulum, the very first cell organelle that we are going to learn is endoplasmic reticulum. So this endoplasmic reticulum, what is it? So endoplasmic reticulum, also known as ER, it forms a network of membrane bound tubes and sheets within the cell. Now this is a network. It is like tangled sheets of it is like a tangled sheet. It forms the network inside the cell and it is always attached to the nucleus. Okay, so here you can see these are some sheets. You can see these are some sheets which are tangled, and it is having sac like structure where we can store something. So this is only endoplasmic reticulum. This is only known as endoplasmic reticulum and nothing else. Now there are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Now what is the difference between these two? So rough endoplasmic reticulum, it appears rough due to ribosomes. It appears quite rough because of ribosomes. What are ribosomes? Now ribosomes are the protein synthesizers. Here you can see this yellow dots, right? So these are nothing but ribosomes. Due to these ribosomes, the endoplasmic reticulum appears to be rough in surface. These are the sites of protein synthesis, and endoplasmic reticulum is the site of protein synthesis where protein is being synthesized. While smooth endoplasmic reticulum is not having these facilities, it is not having ribosomes and all. It is like totally plain, and it is the organelle which synthesizes lipids, enzyme, and the hormone production. Basically, which type of hormones, like the testosterone and estrogen. So steroidal hormones. Okay, steroidal hormones are there inside. I don't know the perfect pronunciation of that. Then ribosome synthesize protein. Ribosomes are here, which are show on it, present on it, and they synthesize protein. Okay, they make protein, by the way. No protein synthesized. So this endoplasmic reticulum, smooth endoplasmic reticulum. Okay, smooth endoplasmic reticulum. It do not synthesize protein. It only synthesizes lipids and fats. Function in lipid production. Not involved in lipid production. Facilitates lipid synthesis. No role in enzyme or hormone synthesis, while function in enzyme and hormone production. No detoxification capability and crucial in detoxification, especially in liver cells. What is detoxification? So it is a term which means to detoxify, to destroy something. So this endoplasmic reticulum, smooth endoplasmic reticulum, it detoxify the harmful poison or drugs which people consume. So it helps in detoxification, it destroys it. Okay, which is present in liver cells. So what is this? Lipid molecules in cell are synthesized by SER, RER, Golgi, plastid. Of course, just now we learnt these are synthesized by smooth endoplasmic reticulum. Next we are having Golgi apparatus. Golgi bodies also. So these act as a very crucial thing for transportation. So this Golgi apparatus are named after a discoverer, Camillo Golgi. So Golgi was a person who discovered this Golgi apparatus. Like he discovered it and he named it after him. Now this Golgi comprises a system of membrane-bound vesicles arranged roughly parallel to each other in stacks known as cisterns. So what it is saying? It is saying that these are the membrane-bound vesicles. Vesicles means the shape. This particular shape is vesicle shape. Okay, so it is membrane bound vesicle shape. It is arranged parallelly to one another, and this thing this thing is known as cisterns. Each stack, so this thing is known as what? It is known as cistern or cistern, anything. Facilitates the packaging and transportation of protein across cytoplasm. So endoplasmic reticulum, rough endoplasmic reticulum passes the protein inside this Golgi body, and the Golgi body help in transportation of the protein across the cytoplasm. Perform the functions such as storage, modification, packaging products of vesicles. So it helps in storage of several things. It helps to modify the things, and also helps in packaging and all, just transportation. Basically it floats in the cytoplasm. It is also known as traffic police of the cell. So this question may come, what is the traffic police, what is known as traffic police of the cell? So you can see Golgi bodies is known as traffic police. And very important thing, very crucial thing is that Golgi bodies play a crucial role in formation of lysosomes. Now what are lysosomes? They are basically known as the suicide bag of the cell. Now we will be learning about it. So these Golgi bodies, these are supposed to form lysosomes inside them. Okay, lysosome. Now let us know what are they. So there is not much about lysosome. Lysosomes are like helping in digesting some things. Okay, so lysosome contains several digestive enzymes within itself. And yeah, lysosome, they are filled with digestive enzymes, acids and all from from rough endoplasmic reticulum. So they are having digestive enzyme which they get from this RER, and they act as the cell's waste disposal system. Means whatever waste is there inside the cell, this cell passes it to lysosome, and lysosome just destroys it. It fight against it and it crumbles it and vanishes it. So therefore known as waste disposal system of cell. They digest foreign materials and worn out organelles to maintain cell cleanliness. So what they do, if any foreign material is there, let's suppose viruses or bacteria enter inside the cell, so these lysosomes will help them to digest when they will die, when we take antibodies and all. And also the worn out organelles, like the worn out means जो टूटे हुए, like worn टूटे हुए जो organelles है, like old organelles and all, so it will just digest it and it will maintain cleanliness inside the cell, otherwise it will create a havoc inside cell. Lysosomes break down complex substances into simpler ones by using powerful enzymes. So this lysosome, they help in the breakdown of very complex substances, and yeah, they break into simpler one and done. This is the work of lysosome. Now you will say Golgi apparatus also do the same. Yeah, both do the same. Now, why this is termed as the suicidal bag of the cell, or the suicide bag of the cell? It is termed as suicide bag of the cell because as they may burst during cellular disturbance leading to self-digestion. Now, why are they called suicide bag? Because whenever any cellular disturbance is there, whenever they think that something is disturbing the cell or any other foreign material has entered, then cell passes all these foreign things inside lysosome, and lysosome totally attacks on them, वो उनके टुकड़े टुकड़े कर देता है, it breaks them all. And also what happen, for example, here are some cells, here are some cells, okay, and one cell is damaged, and here these are the cells, and inside them one cell is damaged. Let's suppose these, some other organelles, some other foreign particles enters inside this cell, then this cell will do suicide. Okay, this cell will only die. Why this will only die? Because it would not infect all other cells. That's why, to cause no harm to other cells, this cell will suicide. And yeah, because cell is already having lysosome will suicide, then cell will burst, and therefore this is known as suicidal bags of the cell. Self-digestion, okay, and involved in breaking down all organic material including bacteria and old organelles. As I told you, it engulfs the bacteria and old organelles, therefore known as suicidal bag of the cell. This question may be asked in your exam. Now, what is mitochondria? Mitochondria is like the very special organelle of the cell. You know why? Because this is only organelle inside the cell. This is only the one organelle inside cell which is having its own DNA. You heard it right. Mitochondria, very special one. Very special one. Mitochondria, the cell's powerhouse. It is known as the ATP generator. Okay. Adeno trisulfate, something like this. So yeah, it is the cell's powerhouse. It is having two cristae, whatever. So this structure, this hollow space inside this is known as cristae. So yeah, it is having two cristae. It has no, this, there is space. It is the name. It has two membranes. It is two membrane bound organelle. Two membrane, okay, two membrane are surrounding it. One this white color, one this yellow color. So here cell organelle, it is the cell organelle which is known as the powerhouse of the cell. It is having two membrane. It is porous, outer, folded inside. Now this membrane white color, which means like I've colored with orange now. So this is the simple membrane, or we can say it is porous membrane outside, and this you can see it is the folded membrane inside. Okay, and the inner folds increase surface area for ATP generation, the cell's energy currency. Now why this is folded? Because it is folded because here it provides more area to the mitochondria to produce energy, therefore known as cell's energy currency. And as I told you, it is having its own DNA, own protein, own ribosomes which produce own energy automatically, because everything is there, like DNA is there, so what they can do, ribosomes are there which will produce protein and all. Mitochondria produce ATP used for cellular activities and mechanical work. So this provide energy used for cellular activities and mechanical work. Okay, they possess their own DNA and ribosome. As I told you, they possess their own DNA and ribosome, enabling protein synthesis inside it, known as cell's powerhouse. Powerhouse of the cell is ribosomes. Next, here we got plastid. Now plastid is a cell organelle which is only found in plant cell. Okay, it is not found in, look here, whenever I try to read this pop-up comes down, I don't know why. This is only present in plant cell, not in any animal cell. Now why, I will tell you the reason. I will tell you everything. So plastid, exclusive to plant cell, exists in two varieties, chromoplast and leucoplast. So see, plastids are the things which are only found in a plant cell, because there are two types of plastid. One is chromoplast and one is leucoplast. Chromoplast means the colorful things. So in human cells, plastids are present, but which chromoplast, not the chloroplast. Chloroplast contain green color, and plant need green color to produce food. But chromoplast, they are having various colors, which flowers appear to be different in color. Why? Because of these chromoplast. And human appear to be various in color, like black, fair color, and these two colors are there, so it is having chromoplast with these two colors specifically. But mostly plastids are found in plastids, that is chloroplast, and leucoplast are found in plants. Okay, so let us read it. Let us read what is written here. First is chromoplast. Chromoplasts are colored plastid which are found in flowers. They are colored plastid having various colors. Chloroplast. Chloroplasts are chromoplasts containing chlorophyll, enabling photosynthesis which regulates atmospheric oxygen level. Now this chloroplasts are chromoplasts only, but with green color only, having only green color inside them. They contain chlorophyll and thus enable the process of photosynthesis and produce food and produce energy and oxygen, releases energy and oxygen in return. And leucoplasts, they are lacking the color. They do not have color. Basically, they lack coloration and primarily function as storage site for starch, oil, and protein granules. So these have no color. They only help in storage of starch, oil, and protein by ribosomes, oil by not oil but starch, or we can say lipid by this SCR. Next we are having ribosome. Just now I was talking about ribosome. What is it exactly? Now ribosome is a membraneless. It has no membrane. Okay, it is a membraneless cell organelle. Here you can see this. Okay, here you can see this green colored thing, and this is only known as the ribosomes. Okay, involved in synthesis of protein. They synthesize protein present in both eukaryotic and prokaryotic. This is only cell organelle present in prokaryotic and eukaryotic cells both. Which now here's the one question in front of us. Which of these options are not the function of ribosome? It helps in manufacturing of protein. This is one function. It helps in manufacturing of enzymes. No. It helps in manufacturing of hormones. No. SCR helps there. It helps in manufacturing of starch molecules. Now see, as per my knowledge, like I'm also 9th grader and teacher haven't taught me. I've just downloaded the PDF now. So as per my knowledge, it produces protein molecules. And here I need to search of this, so I can't tell you the fair answer. We need to search it. Okay, because I can't tell you the wrong answer, right? Even I'll search about it. Vacuoles. So what is vacuole? Vacuole are the storage sacs inside the cell. These are the storage sacs present very bigger in size inside the plant cells. Okay, so vacuoles, they are the storage sacs inside the cell which are small in animal and quite larger in plants. Cell central vacuole occupies about 90 to 50% of cell's volume. So inside the cell it occupies a lot of space, about 50 to 90% of volume, and it is filled with cell sap. It's a liquid fluid. What is the function of vacuole? So function of vacuole is to provide turgidity and rigidity to plant cell. Turgidity and rigidity means what? Rigid means to be rigid, to be fixed, and turgidity means it provides a particular shape to the plant and stores essential substances within it. It contains what it has amino acid, sugar, protein, and in single cell organelle consumed food items. So this is having amino acid inside it, means protein it is having sugar and proteins, and in single cell organelles consumed food items, and in single cell organelles having only one cell organs like amoeba. In these types of organelles vacuole is present, very, like, it is so massive inside them. Okay, it is so massive. So here it helps to engulf the food, or it helps to store the food. Specialized roles: expelling excess water and waste from unicellular organism. Now what is a special role? It gives out the excess water, means expels the excess water and waste from unicellular organism. बाहर फेंक देता है ये. It expels. And also one important thing is that this membrane of vacuole, this membrane is known as tonoplast. The outer membrane, tonoplast is the outer membrane of vacuole. These are known as the storage sacs. And who produce, to remember. Now this is our slide in which we have to remember the basic thing, that who are having double membrane, who are having single membrane, and who are having membraneless. So double membrane bound organelles having multiple membrane, double membrane, two membrane, like one this and inside there one more membrane. So it is nucleus, mitochondria, and plastid. So see, nucleus is having two membrane. We learnt about it now. One inner nuclear membrane, one outer nuclear membrane, and the space within this is known as perinuclear space. So it is having two. Mitochondria, as I told you, one is a crysty something inside, which is in this shape, and other is what? Other is covering like other is one covering like this, and one is this membrane, which we have already seen. Next is what? Plastid. Just now we saw they are also having two membranes here. One is the outermost and one is the inner. Leucoplasts are having more nicely. We are able to understand easily more. Next, vacuole. Now here to remember these organelles, single membrane organelle having only one membrane. How to remember this? Endomembrane system. Yeah. So in endomembrane system, who are working there in endomembrane system? This endoplasmic reticulum. Endoplasmic reticulum is producing ribosomes. Golgi apparatus is transporting ribosomes. Lysosomes are what are they doing? Yeah, they are digesting the extra material, they are storing it inside, and this vacuole helps in expelling means to throw out the waste product. So this is how endomembrane works. So you just have to learn whatever things are there working in endomembrane system. Okay, and membraneless ribosomes and centrioles. Centrosome, there's no detail or there's no thing about centrosome in our class 9, so we are not gonna learn that. Feature plant cell and animal cell. So this is the differentiating table which differentiates between plant and animal cell. So see, cell shape. So cell shape can be square or rectangular in shape. This is irregular. Cell wall is present here, cell wall is absent. Why? Because plant need to be at one position, it is stable at one particular place, so it don't need to move, like it can't move. Therefore, to protect it from the sun rays and all, this cell wall is there. Then cell membrane, plasma membrane, present in both. Then endoplasmic reticulum, present in both. Nucleus, present in both. Lysosome, present, like here they are very low because they do not need to suicide themselves, they do not need to store product and all much, they are already having very big vacuoles, therefore sometimes, and here always present. Golgi, present in both. Cytoplasm, present in both. Ribosomes, present in both. Plastid is only present in plant cell but not in animal cell, because animal do not need any color, green color and all to do things. Vacuoles present in both, but in plants they are quite larger, and in this animals they are smaller. Cilia is what? For example, this is a bacteria, and this type of thing which helps in locomotion of bacteria, this is known as what? Cilia. So cilia is absent in plant because they don't need to move now, but present in unicellular organism, mostly amoeba, not amoeba, paramecium, not amoeba, paramecium, and other things like one was there na bacteria they are having flagella. Okay. Mitochondria, present in both. Mitochondria is present everywhere but fewer in number in plants and numerous in animal. Mode of nidification. Now see, this in 7th standard only we learned plant make their own food, therefore they are autotrophic, and we do not make our own food, therefore we are heterotrophs. Now cell division. Now this is our last topic of the class 9's fundamental unit of life. Cell division. So how cell divides. So there, cell division is the process in which new cells are formed by one organism. Organism forms new cell. This is only known as cell division. Now there are two types of processes. Okay, first is meiosis and then mitosis. So see, new cells formed for growth, cell replacement and reproduction. The two main types are mitosis and meiosis. Now see, first we are having mitosis. This is mitosis and this is meiosis. What is the difference between them? Let us understand. So see, here is the differentiating wala table. So instead of directly telling you about this table, I will show you one diagram here. Okay, let me maximize the space. Okay, now here I will explain you meiosis. Okay, let us understand meiosis first. Extremely sorry for the inconvenience. There is a lot of problem with this. Okay, first we are having meiosis. Now what is meiosis? Meiosis is the process, it is known as the reductional process. It is the reductional process. Now in this process what happens, for example, here is like inside the cell chromosomes are there, right? So here is the one cell. Here is one cell. This cell is having let's suppose 12 chromosomes inside it. 12 chromosomes inside this. Then while division, what will happen, these 12 chromosomes will be halfly divided into 6 chromosomes in each cell. In this cell it will be 6 chromosomes, and these these two will perform mitosis again. This will divide themselves into 2 more cells. They will always form 4 daughter cells. Okay. Okay, and here each one will get 6. Here 6, here 6, it will get 6, So reductional method. Here here 12 chromosomes are divided into 6 each. So therefore reduction is there na, because it is divided. This 12 is not given, it is divided. Chromosome ऐसा होता है ना, ऐसा कुछ. So ये टूट गया है, मतलब ये टूट गया है, and gives the 6 to each and every this each and every cell, and they always form 4 daughter cells. Next we are having mitosis. Now don't judge by the spelling of mitosis, it might be quite incorrect or correct. Mitosis. Now what is this mitosis process? In this process, one cell is there with 12 chromosomes and two cells are divided, one and the other. Here the same number of chromosomes are passed inside both daughter cells. Therefore this is known as what? Mitosis. This is the equational process, and that one was reductional process. So it was the difference about both. Now see here, meiosis. It is an equational division. Just now I told you it is an equational division. Then here it is the reductional division. Here two daughter cells, in mitosis two daughter cells are formed. In meiosis four daughter cells are formed. In meiosis daughter cells have same number of chromosomes inside them as parent cell, and daughter cells are having half of the number of chromosomes as compared to parent cell. It helps in growth and repair of injured tissues. Now why this is having meiosis, why these cells are having whole number of chromosomes? Because these are mostly there which help in repairing the injured things, like repairing tissues and all, scars and all, and this helps in the reproduction of gamete. Okay, then here in a brief state, what happens when dry apricots are left for some time in a pure water and later transferred to sugar solution? Now see, apricot means what? Apricots means what? Akhroट. So अखरोट बोलते हैं ना. So then if they are left in water, it's okay. Then we take it out. Then they are kept in sugar solution. Now see, sugar solution. If we take it out, and this is a container having sugar solution, we kept this acrode inside it, apricot. Here it is having high concentration of sugar solution. Apricot is this much in size, so high sugar solution. It is in hypertonic solution. So if the cell is in hypertonic solution, then what will happen? The cell will shrink. Therefore we will say that the cell will shrink. Then answer will be, if they are kept in sugar solution, then they will shrink. A red blood cell is kept in concentrated saline solution. Saline means concentrated. It is concentrated, guys. So it is also hypertonic only ना, ज़्यादा है उसमें saline, ज़्यादा है, concentrated है, and we kept our cell, red blood cell inside it. So of course it will shrink only. Why will it shrink? Because it is in hypertonic solution. It is in hypertonic solution, so it is obviously it will shrink. Next, what the question is there? What's question is there? The plasma membrane of cell breaks up. What if the plasma membrane ruptures? So all the cell organelles which are present inside it, they will come out in case of animals, but in case of plants it won't change anything now, because plant is already having cell wall as a covering. So the royal leaves are boiled in water. First take the drops of syrup is put on it. So here leaves are taken, they are boiled, and then drop of sugar syrup is there. So see, when the leaf is boiled, then all the organelles will die, like all organisms in the cell will die. So if the drop of sugar syrup is put on it, it won't make any difference, maybe, according to me. But still you can, I don't give you the guarantee of the answer from this question, because I just said something. Golgi apparatus if Golgi apparatus is removed from the cell, so if transportation won't happen, modification won't happen, storage won't happen, so many things will not happen when Golgi apparatus is moved. Okay, answer is there. If boiled royal leaves with sugar syrup, no osmosis due to cell death, demonstrating selectively permeable. Golgi apparatus protein. So here are the answers to your question. You can see them. Now here we are at the end of our chapter. Okay, this was the part 2, and here what mitochondria is conveying message. So mitochondria is saying what it is trying to say. Just a minute, guys. So be powerhouse of your life. Mitochondria is saying that we have to be powerhouse of our own life. Mitochondria. Why mitochondria is saying like this? Because mitochondria is the powerhouse of its life, right? Its cell. So it is saying that we have to be powerhouse of our own life by mitochondria. Really so good thought, so good by mitochondria. So hope you understood this chapter. If you like the video, don't forget to subscribe my channel. Okay, and yeah, here I'll be coming with such videos like this. Subscribe, guys. Bye.